Hi folks, me again. A little tutorial all about the nephrons of the kidney. Hopefully you've seen a diagram of the kidney, and you'll know that each kidney has thousands of these little tubes called nephrons. Holding the phone today, so it's a bit shaky, but it means that I can go in and show you the different parts of the nephron. So there's the Bowman's capsule, and at the inside the Bowman's capsule on the other side is a knot of capillaries. This is called the glomerulus, and things that are inside the blood capillaries here get squeezed out and go into the first part of the nephron. And then what happens is the things travel through the nephron, and some of them get reabsorbed here. We'll talk about that later. And then they go all the way through here. And basically these are things that you want to get rid of from your body, like urea, and the urea ends up being passed out of the nephron. It goes actually towards the pelvis of the kidney and then into the bladder where it's stored as urine. Okay, so for your exam, you just need to know the parts of the nephron and what happens there. So I've got them written down here, and then what we'll do is in subsequent tutorials we'll talk about each bit in a little bit more detail. So number one, I alluded to this earlier, number one is filtration, and this happens here. And the filtration is where pretty much everything, apart from some big proteins and the red blood cells, get filtered out of the blood into the capsule. And when it goes into the capsule, it forms a liquid, and we'll refer to that liquid as a filtrate. Okay, so I've already told you about these capillaries here, and I think it's worth saying at this point that there are capillaries all round the nephron tube, and that's important to know for the next bit as well. So the next bit that happens is at the proximal convoluted tubule, which is right next to the capsule. So basically what's happening is at the proximal convoluted tubule, or the PCT, is all the things that were filtered out here that were useful, like the glucose, the amino acids, the ions, they have to be reabsorbed back into the blood, which the blood capillaries are up here. Yeah. Okay, and that leaves things like the urea stays in the filtrate, that doesn't get reabsorbed back in, but things like glucose is needed, obviously, it needs to go to the cells for respiration, amino acids are needed for the cells to make their own proteins, and so on. So they need to go back into the blood, and that happens at this point here. And then at the loop of Henle, what happens is something called concentration. What we're doing now is we're concentrating the filtrate. We're setting up conditions around the loop of Henle, which we'll discuss in a little separate tutorial, to make sure that water can be reabsorbed. And of course, if you reabsorb water, then anything that's left will be a solute, it will be more concentrated. Okay, so the final thing that you need to know about is what happens here, and in particular here, the collecting duct. And what happens here is water moves by osmosis back into the blood here. And of course, water is essential to our body. And so we don't want to get rid of it all in our urine. We want to reabsorb as much, recover as much of it as possible. And that's what happens at the collecting duct. Okay, so hopefully the camera wasn't too shaky, and we'll look at each of those stages in a little bit more detail in a separate set of tutorials.